Welcome and thanks for joining us for another exciting week and afternoon of MSKCC Science Spotlight. I'm Andrea Schietinger and I'm an assistant member in the immunology program at Sloan Kettering Institute. And as you may know, these seminar series features lectures from leaders in basic and translational biomedical science here at MSKCC. So if you have questions during the talk, you can email them to the Office of Science Education and Training at oset@mskcc.org or by tweeting at MSK Education, and then we will answer them during our Q&A session after the presentation. So I'm delighted to introduce Dr. Dana Pears, our today's seminar speaker. Dr. Pear is the chair of the Computational and Systems Biology program at Sloan Kettering Institute, and she is the scientific director of the Metastasis and Tumor Ecosystem Center at Memorial Sloan Kettering. Dana is a pioneer and a world leader in single-cell data analysis, developing computational approaches and tools for the single-cell space, which have led to remarkable discoveries on cell heterogeneity, differentiation, and fate choices during development, tumorigenesis. And we are excited to hear about her latest discoveries today. So the title of her talk today is A Single Cell Lens into the Roles of Cellular Plasticity in Development and Cancer. And so welcome, Dana. Thanks for being with us today to this seminar. Normally, I would say the stage is all yours, but in these days, we have to rephrase, and I'm going to say the screen is now all yours. So welcome, and yeah, please, Dana.
Hello, everyone. Thanks for joining. I hope you're all feeling well. I hope you're all keeping safe and healthy and not climbing up the wall. I wish you were all here with me because I'm a little bit lonely here at my desk and I can't see you. But I will do my very, very best to try and entertain you in these times and maybe say something scientifically interesting. So this is my disclosure. You know, my only conflict is my very, very strong scientific opinions. And so what I wanted to talk about today is heterogeneity and specifically plasticity. And, you know, some of the earliest work in my lab in heterogeneity had to do with the CyTOF technology and really the ability to begin looking at many cells. In this case, we looked at 16 million cells across a cohort of pediatric AML patients using a single cell technology called CyTOF. So that allowed us to really measure something like 31 markers simultaneously in the same cell. And what we found is incredible heterogeneity, not only between the patients, but actually within the patients. And what you see here in this landscape map is that, you know, every color is a different individual or patient, and you can see incredible heterogeneity within each individual patient, and actually heterogeneity that was very similar and between the patients. So actually the real heterogeneity was inter-patient heterogeneity. And the blue dots here are actually normal cells. And you could see here really this path of maturation of hematopoiesis from the hematopoietic stem cells all the way to sort of more mature monocytes. And you could really see this derailment from regular development in the AMLs, but again, in a reproducible, constrained way across the different patients. And we also did some additional experiments and found that this heterogeneity, this phenotypic differences between the cells within each patient is driven by epigenetic rather than genetic factors. We sorted these populations and saw that their genetics was the same. And really the genetics sort of influenced the palette of choices that they took, but not the actual differences within each patient, within each population. And we see that again and again. And here's an example from my colleague at the Broad by Patel and Tirosh looking at glioblastoma. And again, once you look at single cell resolution, instead of having these subtypes such like proneural and classical and mesenchymal, you really see that each individual tumor is a mixture of all these subtypes. And what we see in the bulk is really the proportions or fractions of these populations rather than a pure single population that defines this heterogeneity. And we find similar findings across cancers. Here at MSK in my lab, we found a similar story in lung cancers and pancreatic cancers. And really, with all these single cell technologies now, really largely single cell RNA-seq as well as single cell ATAC-seq, we really see an incredible, staggering heterogeneity. And I could probably run an entire year-long seminar series with many of my guests and colleagues and still not fill up everything that we could say about heterogeneity. So I want to focus on plasticity. But before that, you know, as you were told, I'm a computational biologist by training. I trained in math and computers. And so I want to explain a little bit more about the data and how we interpret it. And I think that if you understand the data better, then you are the ones who can make the discoveries rather than me, because many of the people on this audience probably know a lot more biology than I do. And really, the main thing we have to understand is the notion of phenotypic space. So we have all these measurement technologies, and we could measure all the different proteins that we can measure in CyTOF, and we sort of have a vector of 30 measurements for the level of each protein. We can take single cell RNA-seq and have 20,000 measurements for the level of each gene, or likewise in epigenetics, the measurement for the each peak. But what we really have is we have the phenotype of that individual cell. We have a vector that places it in this space. And what we've learned is that this space is a very, very structured, well-formed, and interesting geometry. And it's nonlinear. So PCA right here is what people started with, and you really didn't get a separation. What we have here is bone marrow, and each cell is a dot colored by a different color. And you see that this bone marrow really doesn't separate well. But when you do nonlinearity reduction, this is a TSNE, there's UMAP, there's many methods, you really get a separation of the space. This is really a way to model the high linear geometry into sort of low linear space. And why is this phenotypic space nonlinear? You really see this in sort of the context of development. Here's a toy example, but you can imagine that this is CD10 and this is CD19 in B cells. And what we see is during B cell development, first CD10 rises, then CD10 falls, and only then does CD10 19 rise. And so, of course, all high throughput data is noisy. So real data looks more like this than this beautiful line that I wrote. And the distance here in true Euclidean space is very misleading, because this dot is before CD10 rose and fell, and this dot is after. And that's why we really want to work with the nonlinear geometry of the phenotypic space. And the way we do that is with graphs. And this is something that we did again together with Gary's lab, Sean Bendel, Elada Vidamir, and Kara Davis. Basically, each dot is an individual cell, and we connect in a graph the cells that are most similar to each other, that express the most similar genes or proteins or whatever measurement. And now this graph captures the geometry. And you can't take big steps. You have to sort of walk through phenotypic space. You have to walk along the edges between similar cells and sort of stay within the data density. And this really allows us to capture this geometry or this manifold and really understand the biology and the geometry of this. So once we have this graph, it's, populations are click in this graph, interconnected components, they become cell types, they become cell states, and that's worked really, really well in a lot of algorithms. And the connections between these cell states, sort of the paths between them, become cell state transitions, developmental pathways, differentiation, and that's really been a very, very dominant force in single cell biology. And another sort of tool that you have is, again, principal components are the biggest linear axes of variation of data, and diffusion components are very similar, except they remain within the data. They understand that the real space isn't the best geometry, and they find the biggest place of variation within the data that stays within the data density. But one of the most remarkable things about single cell data is the asynchrony of cells. This is what really gives us the power. This is what makes single cell data so powerful and so much fun, in my opinion. And that is that because the cells are asynchronous, if we take one sample from the bone marrow, we get all of hematopoiesis. We get the stem cells, we get the progenitors, we get the developing monocytes, the developing megakaryocytes, the developing lymphocytes, developing dendritic cells, as well as every transitional state in between. And we get this all from a single sample, and we can get it from a human without interventions. We can get it, from patients, and that's really wonderful. And this asynchrony is so powerful that we can get dynamics, we can get regulation. Here, in another example, we managed to look at the transition of epithelial to mesenchymal transition. We could pick up the temporal order of all the transcription factors and chromatin modifiers as they changed, and infer a regulatory network between transcription factors and targets, which we could validate very accurately. 268 TFs out of 292 were validated by ATAC-seq, showing you just how powerful the dynamics is in a single snapshot of single cell RNA-seq. Of course, it's really important to get a good algorithm. And a lot of what we do in the lab is working really, really hard to take these concepts and turn them into very accurate and very robust algorithms. And that's most of the work in the lab. And in this one case, looking again at B cell development, I was talking about this paper earlier, we were able to identify a tiny, tiny population, 7 in 10,000 B cells. And this is where VDJ, DJ rearrangement occurs. And because we were measuring so many dimensions at once, we could get not only the functional characterization of this population, that's where VDJ rearrangement was occurring, but also that we could see a rewired signaling. We could see a change in the signaling between IL-7 and STAT5 that was different just in this tiny, tiny checkpoint population. Of course, this required the accurate algorithms that we worked hard to develop, and also deep profiling. Very many cells. I know it's sometimes expensive to collect this data, but the more cells you have, the more powerful you have, more power you have to really drill down on these rare and interesting populations. And just to show you how rare this population is, this is the full pie of the bone marrow, and this tiny sliver right here that you could barely see, this red sliver, is the more hematopoietic early progenitor cells, and the populations I was just talking about, POP3, is a tiny slice of this tiny slice right here. But getting this rare population and understanding healthy B cell development, we could now also understand leukemia. We could pinpoint this population and now map it. This was, led in Cara Davis's lab, map it to pediatric ALL and find that this checkpoint and this population is really what goes awry in ALL, and also what really drives recurrence of ALL. So again, getting a very accurate map of the healthy system really allowed us to better understand disease and what goes awry in disease. Now I want to tell you about work, from a fabulous postdoc in my lab, Manu, and this was sort of like at the beginning, and here's a much more late algorithm to really look at cell fate choice and branching trajectories, and really understand not only the progression of differentiation, but also a little bit about how cell fate choices are made, when and how. And the idea is that not only is development continuous rather than discrete stages, not only do proteins and genes not just snap on and off with instantaneously, but also the decision to take one cell fate or another is continuous. And over time, as rising and falling and changing their balance, we have different probabilities to reach different terminal fates. And this too is a gradual process which we can model continuously. And so again, this graph I was telling you about, this graph that connects between similar cells and allows us to traverse the phenotypic space between very similar cells, allows us to look at paths of possible differentiation. So of course, if you have a progenitor cell, you can go from the very top to each of the terminal states, and you have a lot of possibilities, a lot of plasticity, a lot of choices. Of course, if you're much more mature, then you have less choices. You're more set along the path. And this graph, and having many cells in this graph and having the full graph structures, allows us to really find all the possible paths and to really count and quantify, how many different paths does each cell have to each terminal fate, and what is the probability of taking each of these paths, and really compute it, and develop a probability matrix of each cell, and what are the probabilities of where it could possibly go. And here I'm going to just give you a little bit of an intuition for this algorithm, and show you that it really boils down to the simplest thing in the world, matrix multiplication. And this is because we can take this graph and turn it into a simple matrix, where in this matrix, you have a cell by cell matrix. Every row is a cell, and every column is a cell. And basically, every entry in this matrix is the probability of a cell moving from one cell to another along one of the edges in the matrix. And of course, the original matrix is built on the graph. So if there's no edge, it's 0. And if there is an edge, it's the weight of that edge. And now, by simply multiplying this matrix, raising it in powers, we can compute where it can go in 5 steps and what probability it has to reach each other fate in 5 steps. So this is where each cell can go in 5 steps along this graph. And now we can power it up and multiply it more and more and see where it can go after 50 steps, where it could go after 500 steps, and where it could go after infinity steps, where it stops once it reaches a terminal fate. And we can see what is the probability of each cell to reach each fate. And we could look at this entropy and define it as a differential potential, pretty much a qualitative metric of cell plasticity. And one of the things that we saw is that there's actually a surprising degree of plasticity throughout development, and cells actually make choices and sort of lose their plasticity much later than we think. And some of these 2D plots that we look at can be sometimes misleading. So we really wanted to apply this to another system, and one of the fascinating systems, one of my great colleagues is Kata Gentinakis, and she convinced me, and I'm now a total believer, that one of the most interesting systems in biology is the endoderm, where this gut tube turns into basically all your major internal organs. And this was a wonderful collaboration between Manu and Sonia, and I think the best thing is in the world when a computational biologist and a true domain expert work side by each other together, and really do amazing biology in a collaborative spirit. And here we wanted to study the emergence of all endodermal populations until the gut tube, and we collected a lot of high quality 10x data. And here are the populations we collected again across different embryonic days. And one of the amazing things, if we look at the embryonic populations, is without too much hassle and too much work, they really organized without any prior knowledge according to a temporal axis right up here, from the earlier time points to later time points, as well as to multiple spatial axes, the proximal and the distal and the anterior and posterior, really, because there's just so much information and organization in the single cell data, both in terms of space and time. But we really wanted to dig into it, and we applied the Palantir algorithm I just described to you and even animated to you to try and decide when, how a cell fate choices were made. And one of the surprising findings of our study is, pretty much for any cell fate decision, and right here we're looking at this high yellow here means that there's a lot of plasticity, there's a lot of entropy, there's a lot of uncertainty in where these cells will go, if they're going to go to the extra embryonic yolk sac, or if they're going to become a part of the gut tube. And we really see that even before there's a morphological change, one day earlier than we thought, at 5.5, the cells still are beginning to make their decisions right here, looking at the degree of plasticity and where it's lost. And generally speaking, if we look at every single stage, we see that cell state is transcriptionally encoded one day prior to us being able to see in the microscope the spatial patterning and the difference, and before the spatial differences begin to occur and be visually under the microscope. Here, when we have the full gut tube, this is at 8.5, we still have a completely smooth gut tube. The entire gut tube, which will later create our major organs, is smooth and looks equivalent. But if you look at the gene expression, it actually arranges in territories, in clusters that really look like the organs that's going to become, that really begin to look like the thymus or the liver or the pancreas or the small intestine. And we could really organize the data without, this is disassociated data. This data, we took the cells and completely disassociated them and completely lost their spatial location. And yet there's so much information in this data that we could do pseudospace and order them along their spatial location, predict which gene is going to, what are the spatial patterns for each gene. And here Sonia did the validation, and we could really see such strong spatial signal in the data that really patterned all these promotal organs, and they already looked like their organs in gene expression space. So the cell sort of knows where it's going. But actually, one of the amazing findings that Kat had found a decade ago is that the cell makes a very early decision to either become embryonic epiblast or extra embryonic tissue in terms of the supportive tissues. And during gastrulation, when the gut tube is formed, these extra embryonic cells mix in with epiblast cells and become a little mixture right here. So we wanted to see what is the, we had some lineage tracing. We had a marker that showed what are the two different lineages, so we could see whether they came from the definitive endoderm or the visceral endoderm, and we wanted to see what are the gene expression patterns of each of these cells. And what we saw is that depending on where these are expressed, they expressed all the master regulators for each of the main organs that are going to come out, the master regulators of the lung, of the liver, of the pancreas, and each cell behaved according to where we had expected. Here are the master regulators, but we could look genome-wide, and we could see that if you look at bulk, the definitive endoderm and visceral endoderm were very different. So if we would have done bulk analysis, we would have concluded that they're very different populations. And that's because their proportion and their distribution across the anterior and posterior is very different, as you can see here. But once you've sort of mapped them into pseudospace, then it becomes very clear, and they're 99% identical. And so you have a convergence depending on where the cells are from these very distinct lineages. We looked at an earlier time point and found something pretty interesting happening. So again, we're looking at these different time points here. Each color is a time point, and this is where the ICM decides whether it's going to become epiblast or visceral endoderm, and you see that this is a very clear decision. You see here the plasticity, the entropy is very high as it decides to become very clearly either epiblast or primitive endoderm with a big split. But here we see another point with high entropy. And what we see is at day 5.5, after the decision was clearly meant, after the lineage is clearly split, we see that again, there's high plasticity, and there is a bridge from epiblast back into extra embryonic visceral endoderm. And what Palantir predicted is that epiblast cells transition into primitive endoderm cells. And this was very surprising, and frankly, our colleagues at the beginning didn't quite believe us, but were convinced to actually do some serious lineage tracing experiments to see if this was true. And indeed, this was in multiple lineage tracing systems, confirmed that cells that are epiblast lineage go to the surface, become visceral endoderm, both morphologically as well as express visceral endoderm markers. And we have this trans-differentiation and really a surprising degree of plasticity. Cells that were meant to be epiblast change location and become extra embryonic. Cells that had been extra embryonic go back into the gut tube and behave similar to the embryonic cells. So really an incredible degree of plasticity in the developing embryo. And what we really see and what I really now feel is that this plasticity further increases in cancer. And just as I showed you before, understanding the healthy system is really critical to understand cancer. Here, one of the things that I'm most fascinated about is understanding this plasticity and really what are the causes and consequences of plasticity in cancer. And what I'm going to tell you here now, in the time that we have left, is a few short vignettes with some more of my colleagues. And the first vignette is, again, a great collaboration with our friends down the hall, Scott, and again, a great pairing between the Rena and Scott lab and Cassandra and mine working side by side. And here we really wanted to look at very, very early stage initiation of cancer. And specifically the idea that many of these cancers, both PDAC And also colon cancer can really be initiated through inflammation. And if you have inflammatory diseases, these can really lead to cancer through some interplay between the oncogenic KRAS and inflammation. And so here we compared the mouse model of the wild type RAS when we injure it and it just regenerates. I mean, our cells and our tissues have a lot of programs that help them regenerate, as well as a model where we have one change, oncogenic KRAS, specifically in the acinar pancreas cell. So not even in the entire mouse, but just in the cell of origin. And so we wanted to compare these again. So we used our favorite tool, single cell RNA-seq. And what we saw is this one single mutation in this one single cell type created a dramatic difference. So here, again, we have a map of the geometry, and we see that there's almost no overlap between the blue wild type cells. I mean, you see this tiny overlap right here, and the red mutant cells. This path right here, we could really see this regeneration program of acinar to ductal metaplasia, but the wild type mouse was much more complex. And again, it's very hard to look at these two-dimensional images, which we can try and learn a lot from them, but really it's taking a lot of very high-dimensional complexity and showing the best it can in 2 dimensions, and that can be misleading because there's a lot of complexity here that we can't see because it's just trying to give us the best 2-dimensional rendering it can, and this is very, very complex high-dimensional shapes. So we thought, okay, we will map these separately. First, let's map only the wild type regenerating pancreas. And here the shape was a lot simpler. Here every dot is a cell. We look at the diffusion component, that biggest component of variation, and we see that there's a clear one-dimensional progression here, a very, very simple geometry in 1 dimension of dominant progression. And this is indeed the regenerative program. Cells have a clear, sort of straightforward, one-dimensional regeneration program trajectory, and they follow it quite nicely. And we see that everything is really well behaved in the wild-type mouse. When we look at the mutant mouse, we see much more increased plasticity and complexity. This shape is already weird. When we start mapping the diffusion components, we see that even 4 diffusion components isn't enough to capture the complexity and all the different paths and dimensions that this data holds. And we looked at the gene programs and tried to figure out what's going on. And because it's already so complex and so plastic, and the plasticity and the choices and phenotypes that the cell can take is such higher dimensional and more plastic, it's really even hard to trace simple trajectories in this data. I'm not going to tell you the full story of this data because part of what I want to focus on is little vignettes of plasticity. But I do want to say that even within 48 hours after injury, some of these incredible diversity of new states that we see begin to resemble even advanced PDAC. So 48 hours in, we see such a dramatic remodeling of the epithelial cells that we already see populations that we later, that are very similar and resemble in many ways populations that are only seen in much more advanced and even metastatic PDAC. Also, we see that this rewiring is largely epigenetics, and epigenetics is a huge player here, and that there is real interaction and synergy going on. So right here, looking at this cluster of epigenetic openings, we see that when both KRAS and injury occurs, we see all these new peaks forming, which remain in advanced PDAC that we see neither in the normal pancreas, neither with injury alone, nor with the mutation alone. But their combination begins to open up these peaks, and there's a lot going on in these peaks. And these are the peaks that remain open all through PDAC. And again, if we look at single cell ATAC-seq, we can see again a dramatic rewiring of this population, and this dramatic rewiring very, very much resembles some of the populations that we see in the transcriptional landscape. So really, there's a great way that we can map both the heterogeneity that we see in the transcriptional landscape with the heterogeneity in the populations that we can see similarly in the epigenetic landscape. I've been focusing on the epithelial cells, but also there's a dramatic rewiring in the immune cells. Again, here is the blue immune cells in the regenerating pancreas, and 48 hours later, the immune cells, and these are healthy, non-mutated cells, are completely different. So the remodeling is also in the immune compartment. And sort of really digging into this story and trying to see what might be driving this difference, we managed to find that IL-33 is really a star of this story. And we see that the KRAS mutant cells are really expressing high levels of this IL-33 cytokine that really plays a big role in tissue damage. And this IL-33 can actually talk with its receptors, both in macrophages, but also in ILCs and Tregs right here. And these same cells that have the receptor for IL-33 release Th2 cytokines. And these same populations that are giving IL-33 have the receptor for these cytokines. So we have a feedback loop between the epithelial compartment and the immune compartment that's really, we believe, driving this incredibly rapid remodeling of these two populations. And of course, in wild type, this doesn't happen. In wild type cells, these cells do not have the Th2 receptor, so there's no feedback loop. These are dark blue, nothing is being expressed. And what we really have is that the active KRAS really makes the communication between the epithelial and the immune compartment go awry and creates a feedback loop that creates this dramatic and quick remodeling, so that after 48 hours, things look so differently. So again, these are not full stories, but little vignettes about just how much plasticity we have in our systems. And so here I want to talk about another great collaboration. One of the things I love about MSK, and I hope you all love all my colleagues there, is that this is just such a collegial institute with people who are not only great scientists, but fun to work with. And this has been a wonderful collaboration with Ashley, who was a postdoc that worked with Joanne and I together. And that's another model where you have one bilingual postdoc who can both do experiments and compute, working across two labs. And here we were looking at lung adenocarcinoma. We also worked with Charlie Rudin, who really supplied us with fantastic clinical material. And we could get these tumors directly from the clinic and see that, first of all, if we take lung tumors, most of the cells are actually immune cells. And you can really, single cells really help us understand that tumors are actually dysfunctional organs. They're not just a bag of tumor cells. And again, just as a sanity check, we can see that when we look at the adjacent normal, we can pick up with single cell RNA-seq cleanly and clearly all the adult expected lung cell types. Things were behaving as expected, everything was working. But when we started looking at the tumor cells, things got a little bit more interesting. In addition to the regular adult cell types, we also got a lot of cell types that are more regenerative, more progenitor-like, that have a role in injured lung. And we really see that these tumor cells are really commandeering a lot of regenerative programs to drive the malignancy. But not only are they driving normal regenerative programs, all these gray cells right here didn't map to any single cell type in a normal or even regenerating lung. And what we saw is that while many of the cell types really matched and mimicked and were very closely mapping to a single known lung cell type or regenerative cell type, many of the cells had mixed lineage. They had many key markers and players and transcription factors and important genes that participate not in one cell type, but in multiple cell types. So we got these very pure lineage cells, but very many mixed lineage cells that within individual cells, within one individual cells, did a mix and match across different programs and different cell types. And we really saw a combinatorial explosion where this plasticity doesn't limit this lung cancer cells to one program, one cell type, one set of abilities, but it really has a much more promiscuous access to all the different gene programs, and really sort of have a buffet of mix and map abilities to really allow it to do things that normal cells have checks. When we went one step further, we saw that metastatic populations were even less constrained. And it took us a while to understand the metastatic populations. Analyzing and interpreting single cell data isn't always easier. But then again, we took our lens of looking through the lens of normal development. And really, that's why understanding normal development and organogenesis, normal embryogenesis, normal regeneration can really help us understand what's going on in the tumor. And what we saw in the metastatic cells is that they were, you could order them along a continuous gradient of maturity. And not only were they expressing programs that really match the programs that we see during embryogenesis and during lung organ formation, but we see them in the same order with the same transcription factors that drive this process in the embryo. We really see that the metastatic cells are mimicking that process using the same tools to attempt to grow organs at the wrong time and place. Digging in, we also saw an interplay with the immune system. We saw that in order to metastasize, they actually switched and found more SOX2 lineage cells. So again, one of the wonderful things is we can't really do experiments in humans, so we can see phenomena, and then we can go to different models, in this case a mouse model, to try and understand what's going on. And what we see is that the cells, when they're disseminated single cells or small incipient metastasis, are really SOX2 lineage cells. And they switch their lineage to SOX9 when they escape to metastasis. And through a long series of experiments, which I won't go into, Ashley really showed that basically SOX9 are a lot less sensitive to NK killing than cells that don't express SOX9. So SOX9 protects these cells from NK killing. And we see the same mechanism and the same cells and the same downregulation of the interferon response in the embryogenesis itself. And really, when we remove the NK cells, we have unconstrained growth, and we see both SOX2 and SOX9 cells in a metastasis. So what we really see is the tumor gaining plasticity and mimicking both regeneration and organogenesis, but with more plasticity, with less control, with less stop, so that it could mix and match gene programs. And really, this too had an interplay with the immune system, and it gained the ability to evade the immune system here and break into a metastasis. So again, in another collaboration, this is really Adrienne's work with a talented student in her postdoc in her lab, Yudan, and really to see, okay, what does this plasticity allow cells to do? How can they really gain the ability to adapt to the metastatic niche? And again, it's this idea that these cells can access programs that they shouldn't be accessing normally. So through a lot of work, and here, kudos to Scree and to Linus and to the entire team and to Vitus for really getting a single cell RNA-seq to work in this very, very hard to work with tissue. It's really important to really optimize the tissue and make sure you're getting all your populations and make sure you're getting them at quality. And once we did that, we could see, okay, what's going on? And one of the things these cells have to do is they have to adapt to a very metabolically depleted environment. There's very, very low levels of irons. And what we found is really all patients from both lung cancer and for breast cancer overexpressed in their metastatic epithelial cells these two iron transport genes at really, really remarkably high levels. Now, if we would have just looked at bulk, we would have thought that this came only from the immune cells, because these are actually binding and receptor genes and proteins that typically are expressed only in immune cells. But here, because these tumor cells had the plasticity to access new programs, they could find the solution, the adaptation to survive in this environment. and find the program that they needed to really survive in this very, very harsh environment. Now, of course, I'm doing Adrian incredible, and you did an incredible injustice by going through years of careful experiments to really show that both these genes are necessary and sufficient for cancer growth. Again, using mouse models, knockdown, overexpression, and many controls to really, really show that these two genes are what allows these cells to grow there. The remarkable thing is not only does overexpressing these genes to much, much higher than physiological levels allow them to take all the iron that they need to survive, they're expressing it so much higher that they're able to take all the iron out of the environment, and now the immune cells can't get the iron that they need to function and to attack them. So in one shot, the cancer cells manage to get enough iron to survive, and manage to deplete all the iron to inhibit the immune cells that are out to get them. And now that we understand this mechanism, this really now finds a vulnerability, a way to target these metastatic cells. And this is working towards a clinical trial, which probably would have already gone and kicked into practice had we not been all suffering from this COVID situation. And since we don't have much time left, I just want to sort of finish with one very quick vignette. I won't tell all the difference, but a story. But again, this story is led by Joe, a fellow in my lab, joint with Charlie Rudin. And this is an entire wonderful team of people. Charlie and Alvaro in his lab, Charles and Wouter in his lab, also Chris and the wonderful rapid autopsy resource, Travis and his magic with multiplex imaging to really understand the drug resistance, again, via lineage plasticity. And there's a mechanism where P53 and RB loss, that's sort of the genetic mechanism, increases the ability of cells to their plasticity, allows them to basically switch lineages to a more neuroendocrine state, in the case of lung, from adenocarcinoma to small cell, allowing it to escape therapy. Here again, in one of Chris's rapid autopsy patients, we could see multiple sites, including a lineage transition from a non-small cell to a small cell lineage. And here you can really see through the MIBI and Travis's wonderful work in getting the MIBI to work that we have two different morphologies. We have adenocarcinoma here, small cell lung cancer here. They are the same origin, they share the same mutations, and you can see that adenocarcinoma is a lot hotter, and the small cell really shuts the immune system down and is a much more aggressive plastic cancer. The same mechanism, the same lineage switching to a small cell neuroendocrine phenotype happens in prostate cancer. And here, together with Wouter and Charles Sawyers, and if you were here, I think it was 2 weeks ago with Charles, he quickly told you this story where we could use organoids to study this lineage plasticity. And I'm not going to tell the whole story, but I really wanted to get to this story again to really show you how you get an intermixing. So at the beginning, we have the basal and the luminal really well split from each other. And as after we induce the mutation, the phenotypes become coming closer and more intermixed and more intermixed. And remember, I was telling you about these graphs and the ability to switch from one state to another. Here's the graphs that match these single cell profiles and the different paths between the cells and the luminal and basal. And you could really see visually how over time the plasticity increases and the cells can just have many new paths and many new choices and basically are less constrained and can go and through multiple paths and switch their phenotype and intermix over time. Because single cell data really gives you the entire genome, we can also see what else is going on in these cells as they're sort of losing their basal and luminal identity and as they're becoming more intermixed and more plastic. And we see a lot of things, but some of the things we saw is an increase in STAT family genes and an increase in interferon. And remember, this is an organoid. There's no immune cells here. This is an in vitro system. And indeed, we saw that JAK-STAT inhibition really restores the luminal cell type. So even without an immune system, these immune cytokines and this immune signaling plays a huge role in plasticity. What I've told you here are a few tantalizing vignettes. We're only at the start of our journey of trying to understand the causes and consequences of plasticity. But what's very clear to me is, recently there was an update to the hallmarks of cancer. I'd like to suggest another update, and this is a new icon that Manu drew for us, and really define plasticity as a key hallmark of cancer. And one of the things that mutations endow cancer cells with is this ability to switch their phenotype and to adapt to new challenges, whether it's to evade immune system, evade therapy, colonize new environments and metastasize. The ability to, this plasticity is an incredible and lethal ability of cancer cells, and one that we really need to understand to really stop a tumor in its tracks. So I'd like to thank all of you for listening to me in this afternoon. And most importantly, I'd like to thank all the wonderful people who did the lab, but both my current lab members, past lab members, and I'm the luckiest scientist in the world to have such a wonderful set of collaborators: Kat, Scott, Joanne, Adrian, Charles, Charlie, and some of the earlier work I showed was with Gary Nolan in the CyTOF. And thank you all for listening. And questions?
Okay, so Dana, thank you very much for this exciting tour de force. So I'm gonna start with a question from a listener with regards to the single cell RNA and expression of genes. So when performing single gene-dependent lineage tracing by single-cell RNA-seq, if the lineage tracing gene is not present in a given cell, where you're only sequencing a fraction of the cell's transcripts, how do you then know if the gene is absent because the transcript was missed by the single-cell technique, or if the transcript is actually absent from the cell and not expressed?
So one of the best ways to do this is actually sort the cells before you stick them. So in the case I showed earlier with Kat's work, one of the reasons we knew that was we weren't counting on the transcript, but we separately sorted the cells and separately ran the cells on separate RANs. So, of course, FACS is not foolproof either and isn't without error. But one solution is to sort based on fluorescent proteins. One of the other things is that because we have such rich signal and because some of these things have such strong signals in the data, we can do a lot of inference. So one, we can often capture the gene of the lineage tracing in the data because it's usually a pretty well expressed gene. So I'd say even if we weren't to sort, we'd get a pretty good capture rate. And because usually the differences are so big, then you can really look at the other genes to do very strong inference. For example, in this case of the lineage tracing with Kat, while I said 99% the cells are similar, there is a very distinct signature that does identify the visceral endoderm cells, and we could use that signal, which involves more than one gene, so we have a good chance of capturing it to really predict in incredible accuracy, like almost 100% where they came from. And usually if your lineage tracing is supposed to lineage trace something with a strong signal, the fact that you're doing a genome-wide expression and the fact that biology is so well behaved, unless you really, I mean, of course, this assumes that you did your cell handling right and your sample is well handled, so your single cell data isn't garbage. But if your single cell data is well processed, then usually biology speaks in a very strong language.
Okay, so then another question is if it is possible to distinguish intermixing of cellular identities between two cell types, and two cell types that are just begin to look more similar by gene expression, by response to common stimulus. So if there are still different cell types, but just by activating a similar new gene expression program.
So again, if we sort of go back to this thing right here, you could see the progression. And again, this is part of the reason I love single cell data. It's the asynchrony. We can clearly see a progression between week 0, 2, and 4. And even within this progression, you can clearly see, for example, in week 2, we have cells right here that are a lot more similar to the healthy and well separated cells, but cells that have progressed quicker and become more aberrant and more plastic, that are more similar to week 4. And what we see in almost any single cell data that I've looked at with any perturbation, whether it's a genetic or some signal or some stimulus or some disease, is that you do have a range. Within a single time snap, you have a range of things that are very close to normal and a range of things that are, aberrant already. And, going back to the epithelial to mesenchymal transition, even 10 days in, we had purely epithelial cells, purely mesenchymal cells, and everything in between. And that ability to look at the transitional cells and how they change and what changes among them, and the fact that this data is just so structured, really allows you to infer a lot.
Okay. So next question is with regards to the organoid model and the interferon signaling.
Ooh, now you're getting me in trouble. Okay, yeah.
How did you come to the conclusion that it was the interferon signaling that was enriched in the p53 RB knockout organoid system, and what was the comparative model to come up with that conclusion?
Again, the comparative model is the pseudotime. This is the power of the system. So what we, and here the royal we is I don't do a thing but talk to you guys and get to present this great science, is because there is this transition here, because we see cells that have very low plasticity and cells that have very high plasticity, cells that are very close to normal and cells that are very intermixed and have mixed programs in them. We can sort of sort the cells along this trajectory from the best behaved to the worst behaved cells and see and now we have, so we have an ordering of the cells along a single trajectory, but each of these cells has a genome-wide program. So we can see what are the gene programs, what are the transcription factors, what are the signaling molecules that change the most dramatically along this progression of well-behaved to poorly behaved or to very intermixed cells. And, pretty much it was quite striking that all the interferon genes, all the STAT genes, all the JAK genes, really, as the cells were losing their plasticity, were just, shooting up. And there's a couple of other things that shot up, but really this pathway, and I'm not showing you all the data, shot up so strong, so consistently, so early in the game across, all the different related genes and also all the different JAKs. And the fact that there wasn't even any immune cells here. And, also you have to trust biological gut. And here it wasn't my biological gut. It was Joe's and Wouter's biological gut to say, hey, this is interesting. Let's try it out. And they were lucky. But why that happens and what's going on there, we still don't fully understand. As I said, I'm giving you unpublished vignettes that are only first part of the story. In the cancer stuff, only the story with Ashley and Joanne is published. All the other things are still very much work in progress, which we're hoping for the labs to open so we can get back at it. Okay.
Next question. Were we to consider plasticity a state rather than a process? Does chaos theory help illuminate the complexity of these processes?
Chaos theory is one theory. It's one set of mathematical tools. And I wouldn't define it as the best math because we actually do want to find a little bit of order in this order. Chaos theory might be able to help quantify plasticity, and actually I've been looking for methods to try and best quantify plasticity, so that's a good idea, thank you. But I don't think it's going to help us with things such as understanding mechanisms, which is something that I'm interested in doing. But I don't think there's going to be one method, one metric, one set of tools, or one tool to rule them all that's going to solve everything. I think we're going to have to attack it from many different angles using many different tools, both mathematical, computational, as well as biological.
Here's a question with regards to the metastasis part and SOX2 and 9. Was SOX2 signature present in all of the mets independent of the site of metastasis, showing lineage transition?
So again, we didn't take, that many samples, but we did have enough to show consistency both in our mouse models and in the humans. And also there's consistency between the humans and mouse. So SOX9 was definitely much higher than SOX2. I wouldn't say that SOX2 was absent as much as SOX9 was really a lot more present. So what we showed in the experiments is that it's not that SOX2 really limits you, but it's actually SOX9 that gives you that ability to evade NK cells. So the goal of these cells is to express SOX9 and evade NK killing, and that's what the again, because it's more plastic, because somehow they've more epigenetic plasticity to access all these programs, SOX9 allows them to evade the NK killing. SOX9, it's not enough to overexpress SOX9. You also have to overexpress SOX9 in this sort of stem-like progenitor state, because it's in that state that SOX9 allows it to access the programs that allow it to evade the NK killing.
So here's a question from a friend of ours. Cell plasticity is key. What should we be doing about it now?
We need to be studying it as much as possible. We need to we need to find better metrics for it, and look in the clinic. I think it's wonderful that we can get things from the clinic. I really enjoy the work with Charlie Rudin and Chris Iacobuzio, where we see so many cases of plasticity happening inside patients, where you can get all these mixed lineage patients. And again, the wonderful thing about the asynchrony is we get the adenocarcinoma, the small cell, and many intermediate steps. But we also need many models, mouse models, organoids models, to understand this process in a more controlled setting. And one of the things I'm most excited about is some work that I'm doing with Scott Lowe, or really being led by Scott's lab, to really allow very powerful lineage tracing to see the plasticity progress in a way where we have CRISPR-based genetic scarring. So we have really all the clonal history as well as the phenotypic behavior of these cells. And I think that once that's fully working to the degree that we want it to, and we're making progress, again, open up ZRC, we'll be able to unlock a lot of understanding through that system.
Okay. And
of course, study the healthy system as well. You cannot understand cancer without really understanding develop healthy embryogenesis and healthy regeneration in our healthy cells.
In the beginning of the KRAS and the IL-33 story, you showed that the IL-33 is secreted by the epithelial cells. Did you see then a shift in terms of the microenvironment, and that other cells within that microenvironment do secrete and make IL-33, and a change in receptor expression?
So again, we looked at this 48 hours in, and also we have another time point 24 hours in, but we haven't looked at that as carefully. So first of all, I have to say that this IL-33 is expressed only in the mutant cells. So the mutant cells really unleash this feedback loop by both causing IL-33 to be expressed, again, through chromatin remodeling, because we actually see the chromatin remodeling that allows for that, as well as the Th2. I believe Direna also did a lot of imaging on that, but since that was really things that Direna's been working on, and I've been really trying to look at the single cell data without the spatial context yet, I haven't really fully looked at that data myself.
And different time points then throughout the progression.
Yeah.
Okay, Dana, there are more and more questions coming in, but we are reaching the end of that seminar series. I want to thank you for this great presentation. I hope you all enjoyed joining us, and we would like to welcome you again on Wednesday, same time at 4:30. Dr. Craig Thompson, our President, CEO, and member in the Cancer Biology and Genetics Department, will be talking about metabolism and cell fate decisions, and I hope you can join us again. Until then, stay safe and well from wherever you have been joining us. Bye-bye.
Bye-bye. Thank you, everyone, and I'll be joining as a participant on Wednesday for sure.
Perfect. Okay. Bye. Bye, everybody.